Good afternoon, Mutograph. I bring you greetings from OPTIMISTICC, one of the other grand challenges, and we're really grateful for Cancer Research UK and some other funders that helped us to get to the grand challenge level. So we have a lay summary here with no conflicts of interest. So we are holobionts. We're composed not only of our human DNA, but also our microbial DNA, which of course is much more substantial than our human DNA. So bacteria on or in our bodies are important in human health and disease. The large bowel contains the richest populations, and therefore it's the best place to look for its effects. These differ in countries with high and low rates of bowel cancer. You do get people surviving into their 70s and 80s in Africa, but they have a much lower incidence of bowel cancer than people in their 70s and 80s in the United Kingdom. Why is that? They have different microbiomes. Is that a relevant factor? And what role do they play in bowel cancer, and how can we use them to prevent, diagnose, and treat bowel cancer? So that's the lay summary of what we're up to. So our vision is a world where nobody dies from bowel cancer. I never thought I'd be saying that, and whether we'll get there, who knows. But our grand challenge is to define the role of the microbiota in bowel cancer, its impact on, or the impact of the microbiota on risk, initiation and progression of bowel cancer, and the creation and testing of innovative microbiome-targeted therapies. And we've been going since the 1st of January this year. So it's a big team from 7 countries, very North America focused. So a lot of Harvard, Johns Hopkins, we have Vancouver, we have Guelph in Canada, we have Hans Cleavers, like most people appear to have. We have Joseph Tabanero from Val Hebron, we have Fiona Parrish from Immunology in Oxford, and ourselves in Leeds. So a big team, but a fun team. And these are our questions. So which, and I'll let you read them rather than me saying them, when, who, where, what, whether, how, and most importantly, why. So lots of work to do. So I, with Cindy Sears, leads Work Package 1, and the aim of that is to map the bowel cancer microbiome. Aim 2 is to investigate microbiome-targeted therapies for bowel cancer, and Aim 3 is to share and disseminate our data methodology. I'll only talk about work packages 1 to 3 briefly, because we don't have much time, and they're the most relevant to this audience. So the overview of work package 1, our goal is to define the microbes associated with increased bowel cancer, both in hereditary and sporadic disease. And the impact of that, or one impact that we're trying to do, is to improve cancer screening and prevention in individuals at risk of bowel cancer. And our approach here is that, led by Cindy Sears, there'll be the hereditary risk survey of 500 individuals with FAP and HMPCC, characterize the microbiomes and the microbiomes of their cancers. And then we're looking at 17,000 samples from 10,000 from the bowel cancer screening program in the UK, 4,900 in our global networks, and Cindy Sears has got a series of 2,100 colonoscopies. And we'll be looking at, as you can see there, hereditary sporadic risks with a number of molecular tools. But a lot of it is initially 16S ribosomal RNA sequencing. And we also have an epidemiological package, which very much is important in the overlap with Mutograph and trying to work together on geographic locations, lifestyles, anatomic location, to understand how this microbiome interacts with the broader environmental exposome, so that we can develop an opportunity to look at the cancer-associated microbiome and intervene in it. Then the third one is also really interesting, which is mapping the cellular location and its interactions with the various constituents of tumours, and what are these bugs doing in these tumours. So we want to understand the spatial organisation interaction of the host and microbial cells in bowel cancer, and this might enable us to identify targets for intervention-based mechanisms, or these host microbe interactions, some of which may be immunological and of importance and relevance. So just to show you some of the early stuff that we have got, we have here, so at the moment we've already characterised, or Caroline Young sitting over there, 1,287 stool samples from the bowel cancer screening programme. There's negatives, positive bloods, but negative on colonoscopy, positive blood, colonoscopy with an adenoma and positive blood colonoscopy on a cancer. And we can see that we have a different profile in the negatives to the cancers, and we see some of the already noted bacteria coming up within our bowel cancer screening patients. And Caroline has identified a top 5, and we're at the moment on the sequencers, there are another 1,200 samples where she is testing these bacteria as to whether they can improve the FOBT prediction of bowel cancer risk. So that will be very exciting because it might be a practical tool, but we will have to take it through into further validation series. But these are bacteria identified also by other people, and so they seem to be fairly consistent pattern in bowel cancer in the literature. We have a global challenge network, which with the OPTIMISTICC network now spreads around the world. And so the global challenge, we built a network: England, Vietnam, India, Argentina, and Chile. We're just adding Moscow and South Africa here. And we've already got 100 microbiomes from around those countries with their 60 controls. And Argentina here, Carlos Vaccaro from Buenos Aires has already not only joined us, but joined you in the Mutographs network, so he's already submitting samples. And so one of the things to think about between our groupings is, can we help each other with these networks to try and get microbiome data as well as DNA data on the same patients, which will be much more powerful ultimately than if we have them in separate patients. So what are the links between the Mutographs network and the network that we're creating here can we establish to help us both get more information on bowel cancer? So these are some of the microbiomes coming out of Argentina. Here is India, here is Vietnam. There's lots of Prevotella in India and in Vietnam. These are nice bugs. They produce lots of short-chain fatty acids. There's a few around here in Argentina, and these are different to what we see in the UK bowel cancer screening program. And so we will be expanding the numbers of cases where we have our controls, but also our bowel cancers. And when you look at the really early data, and this is really, really early data, we have these are from the cancers, we have some significantly enriched in healthy volunteers compared to CRC patients, and these are significantly depleted in healthy volunteers compared with CRC patients. And what's so interesting from this early data is these 5 bacteria are the 5 bacteria that we chose out of the bowel cancer screening program. So we may be seeing the same bacteria in cancers in Vietnam, a very low incidence area, cancers in India and Argentina and Chile, as we see in the UK, but we can see that their background microbiomes are different. Whether this holds up in larger numbers, we don't know, but we'll be very interested to see. Now, Henry Wood, who's our bioinformaticist, also in the audience, has access through the GSIP to 1,800 cancers and the 1,800 germline times 60 whole genomes done in the 100,000 Genome Project in the UK. Genomic analysis has been done by Ian Tomlinson, that's across the top, and Henry has done the microbiome analysis here, which is down the side. And of course, you can't read those, so I apologize about that. But this is being developed, and we've got another 1,200 to be released possibly in the next month or two, and they will be a test set to ensure that we're getting exactly the same data out. And then ultimately we'll be able to do the associations between the driver mutations and the microbiomes and potentially the mutational profiles. So again, very interesting data, which would be very nice to compare to the data that you're pulling out around the world to see, do we have the same bacteria in these cancers? How do they differ? How are they the same? And what is the role of these bacteria? We've also been doing some shotgun low-resolution sequencing of formalin-fixed paraffin-embedded material. These 3 are trials. MRC-007 is a trial of short-course radiotherapy, that's 5 days, you resect after a week, versus none. And what we can report from these 470 cases is we can't see any relationship between the microbiome and radiotherapy treatment. Here we have Quasar, which is a stage 2, stage 3, no preoperative treatment. We have data there, and we have data on Foxtrot, where we've been giving people chemotherapy preoperatively in a trial, and again, we'll be looking at those microbiomes. We also have a GIFT autopsy series, but this is 1,130 samples, and you have to be very careful with the microbiome because these weren't rapid autopsies. So that is under analysis. What we'll get out of that, we're not certain at the moment, but we will at least do them because we have large numbers of metastases there. And the metastases are really interesting, and that's why we've gone on to do another series of liver metastases. And we also have an extreme phenotype, so small bowel cancer is rare. And so we're looking at that extreme phenotype to see, do we have the same bacteria in those small bowel cancers, despite the fact the small bowel has a much, much lower level of microbiome? So again, very interesting to get the answers out of those. The cellular location of the microbiome is fascinating. So Susan Borman showed, you can see Fusobacterium nucleatum in colorectal cancers. This is an antibody to Fusobacterium nucleatum, and on the surface, Fusobacterium nucleatum are very thin filamentous organisms, but when they go deeper, they become bacillary. So they change in morphology as they go deeper into the tumour. What the effects of that is, we have no idea, but we will be Emma Allen-Vercoe from Guelph has developed antibodies to enable us to look at that. There is work going on in spheroids, but also cell culture, which show that the Fusobacterium nucleatum actually invade into the cells and become intracellular bacteria. What's the influence of that? Cleavers has already shown that actually he can demonstrate Fusobacterium creating DNA damage in his organoids. So again, we can see DNA damage. It'd be interesting to see the patterns that DNA damage creates in the DNA. So, benefits for Mutograph could be additional Mutograph sites, if you're interested. Is the mutational signature seen in Mutographs associated with the microbial signatures in different countries? We will ultimately have up to 3,000 UK bowel cancers from the 100,000 genome, which will provide some very interesting data, and working with you on some of those mutational signatures might be very interesting. And we ultimately hope to get a genotype microbiome phenotype analyses as we hope to collect the bowel cancers from the 100,000 genomes in order to look at the phenotypic impact of these microbiome profiles. Benefit for us in OPTIMISTICC is if we can extract microbiome data from all the Mutograph bowel cancers from all your submitting sites and compare that to the mutational signatures, that would be unique data we would otherwise not be able to get. Comparison to the whole genome sequencing microbiome data from the UK and any OPTIMISTICC sites with whole genome, and then the association of microbiomes with mutational signatures. Are some of your unknown mutational signatures bacterial in origin or inflammatory? And we need to know, do we find the same bacteria in the cancers in different parts of the world? Because do we have to change screening in the UK versus Vietnam or Argentina? Do we have to change therapy or treatment? And there's evidence that the microbiome interacts with therapies. And it would be nice to collaborate with the Mutograph sites to enhance OPTIMISTICC as well as to enhance Mutograph. So in summary, all bowel cancers have a microbiome. We forget this, but it's there. The microbiome of patients with cancer and polyps differ from those without in the stool. This may aid screening. But we do not yet know precisely the microbiome's role in causation, promotion, CRC immunology, response to treatment. And we do not know if modulating the microbiome is beneficial. We hope that it is for the patient's sake. So we hope that you can help us, and together we can both get to the answer. So here's the Leeds team, Southern Hub, who's given us all these screening samples, so 2,700, the OPTIMISTICC team, and our Grand Challenge network, of which Carlos is within the audience. And so just to say thank you, Mutograph, for last night, for having us here, and we hope that we can help you, and we hope you can help us. Thank you.